I can't hear. Post the exam answers, or? The previous exams are posted on Blackboard, right? That's part of the study guide. Right? Everyone gotten hold of the study guide? Very important. Look at those previous exams. Use those exams to test your knowledge. This exam will be quite similar to previous exams. Okay? So it's posted on Blackboard as the study guide, and it has the last several years of exams with the answer keys. Okay, any other questions? Okay. So last time, what I'd like to do today is finish talking about signaling and growth control, this new type of signaling pathway, the elucidation of this signaling pathway in large part owes cancer biology, cancer genetics studies, and biochemistry, cell biology, to come up with novel modes for attacking a signaling pathway. And last time I talked about the identification of the first oncogene, the first transforming gene that was found in what? Anyone remember where this gene came from? Yeah. Yes, good. There's a virus that infected chickens, Rous sarcoma virus. Harold Varmus, Mike Bishop, isolated the gene, demonstrated that that gene was both necessary and sufficient for transforming fibroblasts into cancerous cells, uncontrolled mitogenesis. And that gene, Mike Bishop hypothesized, was a mutated form of a cellular gene that through evolution the virus picked up and mutated, and when reintroduced into cells in a dominant way caused cell transformation. And he showed that there was a normal cellular gene, which he called, so the viral oncogene was called VSARC, viral SARC gene, and the normal gene was called CSARC for cellular SARC, the normal cellular gene that is not mutated, not transforming, and presumably plays some normal role in the cell growth control pathway. Okay. What you see here is a three-dimensional structure of the normal cellular SARC gene. The viral SARC gene is an oncogene. The cellular SARC gene we call a proto-oncogene. Okay. It is not oncogenic, but it gives rise, in the case of a virus, it gives rise to the viral gene, which is an oncogene, and you'll see other ways by which this gene might become oncogenic. Okay. So it has several interesting, its overall folding has several interesting components that give rise to the folding of this protein, and we're going to talk about that in this lecture. It contains the catalytic domain, which we'll discuss in a few moments what that catalysis is. It has an SH2 domain, which stands for SARC homology domain, which plays an important role in many proteins that are involved in signaling processes, and we'll see how. An SH3 domain, as well as an activation segment. So all of these terms indicate some kind of functionality, right? And it's important that we understand it's one thing to isolate the gene and isolate the protein and even crystallize the protein and understand the structural configuration of the protein, but it's a whole nother thing. What we really want is the functional significance of this protein. What is the functionality of this protein? What is it about the viral SARC gene that when introduced into cells causes uncontrolled growth? And what kind of functionality does cellular SARC have in normal cells in growth control? The first clue to the functionality, really the first demonstration of a functionality for the viral Sark protein, came from Ray Erickson's lab, in which he purified the protein that was synthesized off of the viral Sark gene, off the virus. He purified the protein, and he put it in a test tube, and he started looking for some kind of activities. And the one activity that he identified came about when he put ATP in the test tube together with purified Sark and a mixture of cellular proteins. And he found that when the ATP was made radioactive, it transferred radioactivity to a bunch of cellular proteins. Okay? So in the presence of ATP, cellular proteins were able to become radioactively labeled in the presence of viral Sark. What does that suggest? Yeah. Yes, excellent. It's a protein kinase that phosphorylates proteins. Okay. That was a major discovery and got him into as a member of the National Academy of Sciences. He's on the list of people that are candidates for a Nobel Prize. It was extremely important because it pointed to the first oncogene as being part of a regulatory pathway. Everything that was known about protein kinases had come really from the work of Krebs and Fischer with cyclic AMP-dependent kinases and the kinases involved in glycogenolysis. And now growth control, a key component of the growth control process, which caused uncontrolled growth, was a protein kinase. Okay, suggesting that protein kinases played a wide role in mediating changes in cellular physiology in a wide range of processes. And the question was, what is the substrate for the Sark kinase? Okay, so it's a protein kinase. We take the viral Sark, we put it into cells, and what is it phosphorylating? And the first indication that this was a unique type, a new class of protein kinase, came from the work of Bart Sefton and Tony Hunter working at the Salk Institute in San Diego. And what they found, they developed a method for separating phosphoserine and phosphothreonine and phosphotyrosine from one another on thin layer plates. Okay. And why would they look at those three amino acids? Yeah? Excellent. Okay. Each of the amino acids, these are the only three amino acids that have a hydroxyl group on their side chain and which are phosphorylatable by a protein kinase. And so they just simply wanted to determine which amino acids it had more specificity for, thinking that it was phosphoserine or phosphothreonine, just like the other protein kinases that had been identified to date. And what they found was extremely surprising. And this experiment shows control cells in which which have been fed radioactive phosphate so that all the ATP becomes radioactive and kinases could transfer their phosphate to proteins. Okay, and then we could detect those proteins that are phosphorylated, and if we cleave those proteins into the individual amino acids and run it on a thin layer chromatogram, we could separate those phosphorylated amino acids from one another, and the ones that show up dark were exposed to an X-ray film because they're radioactive. So the radioactive spots indicate where P32, where radioactive phosphorus is located. And in normal control cells, you see that phosphoserine is the predominant, followed by phosphothreonine. So in normal cells, there's a lot of phosphoserine and